Let's turn to another agent that was presented in the same session and has clear, encouraging evidence for efficacy in acquired resistance to EGFR mutations, and that is the Clovis compound CO1686. Dr. Alicia Sequist presented that data. Can you, Nasser, tell us a bit about the highlights for that?
Well, that actually I found incredibly compelling. It is a very active drug in patients with the T790M mutation. It's very active, and similar to the AstraZeneca drug, it also spares the EGFR wild type, and it seems to spare it to a greater extent. So very active drug after patients have already progressed on an EGFR TKI first line. I think the one concern about that drug is a different side effect that was noted in a large number of patients, and that was hyperglycemia. And to the extent where they needed medication for it, and I believe it was 50%, which is a substantial number. And one can sort of guess as to why that's a major side effect. Many think it may be because it has some interplay with the insulin growth factor-like receptor, because we know that class of drugs causes a significant amount of hyperglycemia. But it was in the majority of patients. It's a substantial toxicity. In fact, much so that Dr. Lynch, who discussed the paper, had sort of half joked but half serious, said maybe we were studying a combination therapy of metformin plus the Clovis drug. But, you know, in oncology, we're so used to modest changes and modest advances that we don't see things like we do with the Clovis drug very often. And I think we're excited, and I don't think it's overly ambitious to say that we should expect these drugs on the market soon. And I do. I expect these drugs on the market soon. But we're going to need to know how to handle that hyperglycemia with the Clovis drug if that one makes it to market.
I thought it was, these two sessions were so great. The investigators are such good speakers, both told a really good story of the development of their drugs. I think I realized in sort of thinking about the two of them, Dr. Lynch's comparison was, that was also very enlightening. The response rates seen in both drugs were similar, but 53% in the AZD drug, 58% in the Clovis drug. Dr. Sequist also showed that while the progression-free survival measurement endpoint has not been reached yet, it is at this point over 12 months, which distinguishes this drug from the other, or the first and second generation EGFR inhibitors, where we think of 12 months being sort of the upper limit of what we expect as a median. Every patient is different, of course. I thought also then to go on to the FDA analysis that followed, suggesting a very tight correlation between objective response improvement and PFS improvement was just a great dovetail for this discussion.